Hello, let's talk poxviruses. So shown along here on the top are some personified poxviruses from history, which is neat. So we've got Guangdong, who is supposedly associated almost mythologically with bringing in public health reforms that led to the eradication of smallpox, or at least control of it in China, sort of medieval China. Shitala Mata from northern India is a deity sometimes associated with smallpox. So this is a thing, rather something you could pray to get rid of the smallpox, or so it would pass over. And Sapona is from the Yoruba people in, I believe, what would be northern Nigeria. And he's got a little broom normally, and the broom is actually for sweeping poxvirus into somebody's house. So you would have a little statue like this to make sure your house is safe. And, if you got somebody you don't like, you might send him over that way. At least as far as I understand it. So what is a poxvirus? So these are going to be viruses that are related to smallpox. And smallpox, the actual virus name is going to be variola virus. And there was the major strain of variola virus and the minor strain, and the major was just more deadly, and the minor was more mild. But of course, your results may vary. Everybody's immune system's a little different. And so it's hard to say whether one is, going to give a guaranteed outcome in everybody. That usually won't happen. The genome is going to be double-stranded DNA, about 250,000 base pairs. And when I was working on my PhD, there was actually a guy in the other half of the lab who was sequencing a poxvirus by himself. Back in the old days where you could get about 500 nucleotides from any one sequencing reaction. And that was about the top. And so, 500 nucleotides times a very large number, and you get out to your 250,000. It is a multi-layered, very complex architecture, and we'll get into kind of how that is made. Very interesting. Variola virus is only currently known to infect humans, the smallpox virus, anyway. And the nice thing about this is that it does produce lifelong immunity if you survive. Of course, that's the thing with smallpox. Not everybody survives. This thing has been very lethal throughout history, has killed up to a third of the people that got infected at different times and different places. The amount varies, but the result is not good. And this would be presumably if they got the variola virus major. So there's another pox virus, and it's called vaccinia virus. And if you've been part of the armed forces, or if you grew up in a different country, chances are you may have been vaccinated with vaccinia virus. And so vaccinia virus is where we actually get the name vaccine from. This was the original vaccine. Pretty cool. And it's the vaccine against smallpox. So this is the one that Edward Jenner came up with back in the 1700s and started off the whole idea of vaccines. The root word is vaca, which if you speak Spanish, you'll recognize as a very similar sounding word to what cow would be. And also in Latin, that would work the same way. Would it? Actually, no. Boss. Yeah. Anyway, it doesn't work in Latin. But we're getting beyond ourselves. Vaccinia virus is what used to be cowpox virus. Or rather, there's this interesting story. So the original experiments take place with what is presumably cowpox virus. And so there's a cowpox injected from a milkmaid's hand into this young boy, James Phipps. And we'll get to the pictures on that in just a minute. I think they're in the second half of this poxvirus talk. But that's how it worked. That's how we got the first vaccine. They were using cowpox, which was close enough that it actually worked as a vaccine against variola virus, the smallpox virus. But of course, remember, this is the 1700s. And this is long before we knew about DNA or modern refrigerators or anything really. And so they started out with cowpox, and they tried to grow up more of it. And they would inject this into cows. They would also inject it into rabbits because it turned out that rabbits could be used to grow the cowpox, and they needed a lot of little pustules because that's what they were taking the fluid from and injecting, and that's how they were making the vaccine work in other people. And eventually, somewhere down the line, somebody finally got this thing and sequenced it and read out the sequence of cowpox as well. And these two viruses are nothing alike. So vaccinia virus is a virus that we use as a vaccine for smallpox, and nobody actually knows where it comes from. Presumably some kind of cattle or some rabbit, maybe even a horse, somewhere back in history, had a virus already, which was probably vaccinia virus, and was given cowpox, and vaccinia virus, shut the door and grew anyhow. And the switch was made, and it still worked. And so that's why nobody ever noticed, because the vaccine was still vaccinating. Everybody assumed it was still just cowpox all this time, when actually it's a very different, distinct virus, vaccinia virus. And vaccinia virus appears to be extinct in the wild. So we don't have any way to figure out exactly where it came from and how it came to replace cowpox, which is what everybody thought they were using. But it turns out that vaccinia is, if anything, a little bit better than cowpox. Usually only produces a mild rash when you put it in. Occasionally can spread if you touch it while it's still active and then rub your eyes, for example. This can lead to ocular vaccinia, or if you inject it into a baby that is already immunocompromised, their immune system doesn't work right, then they can end up with full body vaccinia, and in both cases this can be very serious, possibly even fatal. So yeah, interesting viruses, vaccinia and variola. This is a nice little overview of the poxvirus life cycle. So this is going to be another one of these big DNA viruses. And whereas RNA viruses tend to go for a quick replication strategy, they try to get in, they try to get out, and they try to, cover their tracks. DNA viruses can go about this in a totally different way. They can take their time, and DNA viruses that are as big as a poxvirus, remember, they still just have to have one polymerase to copy themselves and just a handful of genes to actually do the real work of replicating the virus and making new virions. And all the rest of those genes are there so that they can affect the host. They want to control exactly what the host cell is able to do at all times. And poxviruses are masters at this. So a virus is going to come in, and there are two forms of this virus that can actually infect. One is called the intracellular mature virion, and the other one is called the extracellular enveloped virion, which is the same as an intracellular mature virion, IMV, but with an extra membrane around it. And either way, if it arrives with or without that extra membrane, it can actually bind to receptors on the cell, and it can get in, and then the core will uncoat. And it's going to make first its early RNAs, which will make its early proteins, which do the copying. And so the first step is going to be make copies of itself. Then it's going to go to its intermediate program of expression, and it's going to make its intermediate proteins. And these are going to change the way the cell works. So they're going to stop the cell from doing apoptosis, which is cell suicide. And they're going to lift any sort of antiviral state that the cell tries to go into. So like coronavirus did, but just masterful in comparison, really. And they're going to change signaling inside the cells in a way that is favorable to the virus and not favorable to the cell. And so anyway, these intermediate proteins are then going to lead to the late transcription. Eventually, they're going to set off the late transcription directly, which is going to make the late proteins. And the late proteins are the ones that will actually package up brand new virions to leave the cell. And so they're going to assemble these virions in the cytoplasm, and then it's going to go up to the Golgi and actually stick to the Golgi and get the Golgi to wrap around it and leave a double membrane. And so this is called the intracellular enveloped virus. And it actually has two extra membranes beyond what the IMV has, because this Golgi stack is going to be wrapped around it. And so imagine you just stretch that Golgi out. See, it's got a membrane on this side and a membrane on the far side, and you stretch it until it goes all the way around. It's still got two membranes, an inside one and an outside one. And then when you have one end join the other, you get hemifusion, then total fusion. And yeah, boom, your virus is now wearing a double membrane coat. And so that's what the IEV is. It's going to go up to the surface of the cell where it's going to merge and do like a fusion. And so the outer membrane is going to leave with this fusion. And so you're going to release the extracellular enveloped virus that only has one extra membrane instead of two. And that's how it comes out. So virions are going to leave by budding, the same process that coronavirus or HIV did. They're not going to lyse the cell. So something like the poliovirus actually will explode the cell once it's made enough copies of the virus, and then they'll go out. But pox viruses, like coronaviruses, like influenza viruses, and like retroviruses, likes to play it cool and take its time. Okay, so this is probably a little outdated, but let's look at the proteins needed for entry. So with all those simple, you remember the simple days of HIV and coronavirus and just one spike protein or GP120 and GP41, and that does the entire job of entry and fusion? Well, vaccinia virus is big, and vaccinia virus has a lot of proteins, and they all work together. So the first one, and so the way that proteins are going to be named in a pox virus is they're going to divide the entire genome up into segments. And they'll do this with something like a restriction enzyme, restriction endonuclease. This is just an enzyme that will cut at a certain signal. You know, anytime there's 6 particular bases in a row, it'll cut. So it's kind of a random way of dividing up a genome. But this is how they did it. And they sorted them out by size. And then for each fragment, because it was easier to work with the fragments, they just started numbering the genes. 1, 2, 3, 4, 5, 6. So L1 is the first gene on fragment L, A16 is the first gene on fragment, or 16th gene on fragment A, etc. You'll have some of these where they will then also mark which direction it is, because unlike all the other viruses we've been talking about, these are DNA viruses, and just like real DNA, they're going to have genes that go forward and genes that go backward. So they don't really have a positive or negative strand RNA. They have some, or DNA rather, they have some genes on either strand. And yeah, that's just how they operate. So sometimes you'll see an extra letter at the end, which is R or L. And so that tells you whether the gene is facing to the right or the left in the arbitrary system that they use for numbering that particular virus. So L1's the first one involved. It's going to attach to a protein called integrin and also a protein called laminin. And integrin is a transmembrane protein that's going to be expressed on the cell surface. And laminin is going to be the protein that makes up what's called the basal lamina. So this is going to be on the sort of underlayer of an epithelial layer. You'll have a layer of laminin. And yeah, I combined both of these at the cell surface. And yeah, if you were to make just integrin or laminin and stick it to the virus, it actually is then blocked from getting in, because L1 has to bind these on a cell, otherwise it doesn't get in. So it's working like a fusion protein, like a normal fusion protein. But in addition to L1 is not enough to get in by itself. It's like an attachment protein. There is this club, and there are at least 10 other proteins. There are 8 proteins that assemble into a complex. And so there's one of each of these proteins in the complex. So A16, A21, A28, G3, G9, H2, J5, and L5. And together, these things make this awesome super complex like, I don't know if you know an old cartoon called Voltron, where you'd have all these little tiny people running machines, and they'd get together and form a big space robot and go, fight battles. This is kind of like a Voltron deal, but it's going to be involved in entry. And if you take away any one of these proteins, then no entry. There's a 9th protein that is not part of this complex, but it will associate with the complex. So you might have 10 complexes and only 5 of these F9 proteins. So that's why it's substoichiometric. Stoichiometric means in the same amount or in a fixed amount relative to the others. So if there's 1, 1, 1, 1, 1, 1, 1, and 1, then there might be 0.7 of these. Rather, there'd be 1 for every, or 7 for every 10 of these other complexes, something like that. The 10th protein, I2, is also necessary, but it's not clear whether or not it forms part of the same entry protein complex. And I actually copied this slide down a couple years ago, and so, yeah, if you were to look now, you might find that they've actually figured out a function for some of these proteins, or even a structure for some of them. So that'll be interesting to read if you end up getting this as one of your questions. Okay, how's it work? So the vaccinia virus, so the main vaccine strains, there's two of them. One's called Ankara, like the capital of Turkey, and the other one is called Western Reserve. And we tend to use Western Reserve in the lab, and for some of these vaccines, there are other places in the world where they use vaccinia virus Ankara strain for their vaccine. But they both work. They're both pretty good and quite similar. So Western Reserve, so this is our, wherever you see a WR, that just means vaccinia virus. So vaccinia virus is going to bind to these things on the surface, and these are going to be glycosaminoglycans, which is just a sugary, proteiny chain, let's say, with lots of branches coming off of it. This is going to be the sort of thing you find on the surface of most cells. It's part of the intracellular matrix. And laminin is also going to be in this area. And so we're actually going to stick to the laminin, and that'll get us associating with these. And it may well be that the virus can bind some of the sugars on the end of these too. Yeah, we would have to look that up. Then anyway, once the virus is attached to that, that's like its attachment receptor, laminin, and then it gets to its integrin receptor. And so this is like the second attachment receptor. So it goes laminin in order to get to integrin, because it's kind of a short protein. And once you have the two integrins that are assembled together, bound to something, this protein called CD98 is going to come into the complex and lock into place. So you'll get alpha and beta integrin together and CD98, and together these three things will cause this entire region with the alpha and beta and CD98 to be pulled into the cell. And then, oh my gosh, the vesicle right here is going to slowly drop in pH as it is acidified because of those proton pumps, just like in every other case. Some of that histidine and maybe some of those proteases in there are going to destabilize or activate the actual fusion proteins. And we're not sure which ones are actually the fusion proteins, if it's the L1 protein or some combination of the others. But we do know that you need all of them for the entire process to work. And so somewhere out there, yeah, is the answer. Somebody will figure it out eventually. And anyway, what it does then is it will fuse with the endosome and drop the insides of the virus out into the cytoplasm. And at that point, it's infected. So it is the virus binding to laminin to get to integrin, alpha and beta. It actually binds to the alpha subunit, but that brings in beta, which brings in CD98. And when those three get together, we're going to internalize all of that, the integrins, the CD98, and the vaccinia. So you just have to imagine the other proteins down here still holding on. And then once the pH drops, then, yeah, we're going to protonate some histidines, and we're going to release the core of the virus. So very similar. Okay, and then once it gets into the cytoplasm, you're going to see another familiar process. So like coronavirus, but unlike influenza virus, this thing is actually going to cap its own messenger RNAs, and poxviruses are very careful to make that perfect cap. It's called a cap1 structure. And so vaccinia will encode its own version of a terminal phosphatase, which remember does the first step. So terminal phosphatase cuts off that extra phosphate at the end of the RNA chain. And then it'll encode its own guanylyl transferase, which transfers guanine. And so it's this backwards GTP, which is going to form the cap. And so there I drew all these triangles going this way, and this one's backwards to show you that's the backwards GTP cap. And then we're going to have an N7 methyltransferase encoded by the virus. And boom, it's going to put that first methyl group on there. Then we're going to have a 2' O methyltransferase encoded by the virus. And boom, that's going to put the second one on there. So just like coronavirus, just like that other process that you now understand very well, the vaccinia virus is going to build its own cap. And so for each of these pox viruses, there's going to be a name. There's going to be a letter of the alphabet and a number and maybe a right or left after it for each of these genes. We've already learned the genes that are responsible for some of the other viruses. I don't think that's going to be important for poxviruses. So this is something that you can look up if you ever want to get into poxviruses, but it's not something that I'm going to expect that you know. And anyway, once we've added the 2 methyl groups, then we have a finished messenger RNA, which also has that secret second methyl group that prevents RIG-I or MDA5 activation. And so the cell does not know that it's infected because the virus does a very effective job of camouflaging its own RNAs. All right, so how does the virus actually copy its DNA? So the virus is a DNA virus. It makes RNA to express its own proteins. It has its own RNA polymerases, in addition to its own DNA polymerases to copy itself. And so let me just show you this, because it's kind of neat. So you've got the end of the genome, and it's got 2 sides, and we call these the flip and the flop side. And there are bits where it doesn't quite bind, so imagine there's base pairing between flip and flop sides, except where there's a little bulge. This is a nucleotide that does not pair with a nucleotide on the other side. And the pattern of nucleotides that are paired and not paired ends up looking kind of like a key, which is exactly what it is. This is our little hairpin, and it even kind of looks like a hairpin, doesn't it? Yeah. And there's one of these at either end of the genome, and the genome is a closed circular DNA, just like that. And so to copy this, there's a couple of steps that the virus actually goes through. So the first thing, the virus is going to wait for a host enzyme to come along and probably force this host enzyme to cut the DNA at a very specific place. So step 1 in this virus copying itself is that it actually breaks its own DNA chain, which, may seem a little risky because I would say it is a little risky. But it's going to break it just before one of the flip-flops at the end, and it breaks it at exactly the same place every time. Uses a very specific way to do this, if I remember correctly. Okay, so once we have cut the single-stranded DNA of the genome, then the DNA polymerase is going to bind to this 3' end, because that's what DNA polymerases do, and it's going to be stapled down to this side a little bit. And so the DNA polymerase is then going to go out and copy the flip and the flop, and it's going to copy until it gets to the end. And so boom, it's just got to the end. Okay, now what? Well, now you just wait for the flip and the flop that it just made are going to fold back on each other, just like this. They'll go boom, right back down. Oops. And once they've done that, now the virus, the polymerase, is at the end of a chain, and look, it has its own little template it can read, and so now it just keeps going. So it makes a flip-flop, waits for those to hairpin together, and then bam, it's now got a new template. It can keep going, and it goes all the way around backwards until it gets all the way to the end, and then once it gets there, it's going to flip-flop again, and it's just going to keep doing this. It's going to make multiple linked copies of the genome, and these are called concatemers, which means one thing sort of stuck onto the end of another and all joined together with no spaces. And so here is one genome up to this point. Here would be another genome up to here. Here would be the start of another genome, etc., etc., etc. And then the last step is that the virus has these enzymes that will actually come in, and it will snip off. It'll snip apart each of the genomes, and it snips them apart at the flip and flop. And once it does this, it will actually join the strands up to make circular DNA. And so you'll end up with circular double-stranded DNA. And when that goes into a cell, we'll peel it apart to single-stranded DNA and go right back here, nick it, copy as far as we can, rather to the end. Flip and flop will boop, flip-flop together, and now DNA polymerase can start reading backwards. It's still attached to exactly the same place, the end of the red one, but once the red one has folded on and is now bound to the purple one over here, then the polymerase has something to copy, and so it does. And yeah, it's that forever and ever and ever. That's what poxviruses are doing. They just make lots and lots of copies of themselves and then chop them up into the right size pieces. And it looks like I need another pair of scissors there. I've, rather mangled that chopping. Thankfully, the virus does a better job with it, though. All right, thank you very much. That's the end of this part.